there is a buzzing noise. Or... Hello?
Yes, Madhumita, you said something?
Yes, sir. There is a buzzing noise.
Okay, 1 second, I'll try.
Okay, sir.
Is there a noise? Can you confirm, please?
It's better now, sir.
Okay.
Okay, sir.
Subhashri, can you do you hear any buzz?
No, sir. Now I don't hear any sound, sir.
Okay, yeah, that's fine then. Sorry, I have some cold, so okay. Good afternoon, everyone. We have kind of last topic we have is genome a genome-wide CRISPR screen identifies a restricted set of HIV host dependency factors. So I will just give a background to it, and then we'll try to look into the actual content or actual point of this paper. So what we are mostly interested is about the usage of CRISPR, and we are reading this as an application of CRISPR. Okay, when we read about CRISPR, we learned that we can do many things. We can delete genes. We can kind of knock in genes. We can knock out genes. We can inactivate. We can also regulate the genes expression, location, and many other things. In that sense, how it can be used is an application like screens. I will get to each of these, and then we also have discussed in one of the last classes about HIV, about gene therapy approach. We learned about some factors, and this study is about trying to find more host dependency factors. Okay, we will get to that. Let me quickly recollect, review what we already studied before with regard to HIV. Every time when we approach the paper, we should identify the subject of the paper. The subject of the paper is HIV. I'm sorry, cyclone, allergic cold, and so on. Yeah, well, so the subject of the paper is HIV. The approach is CRISPR, a screen based on CRISPR, a genetic screen. Okay, sorry. So HIV is a retrovirus. It has RNA genome. It is enveloped virus. It infects T4, CD4 cells, T cells. It has reverse transcriptase, which will make it cDNA of it, which will then go and get integrated into the genome. It can also produce more number of RNAs, and also through translation of them, it can produce all the capsid and code proteins. Sorry, the envelope proteins, and then new virions are released, which will infect many other cells. So through the story of Berlin patient, we have learned about one of the mutant or the variant. It's about CCR5. The HIV requires certain factors for adhesion of the target cells and then being able to be taken up by the target cells. On its surface to make specific interactions with the target cell. And the target cell has certain factors, something like these. These are the host factors that are required for HIV to infect it. So there are two things. One is the HIV should have certain factors, like here it is GP120, GP41. Those belong to the HIV. And there are CD4 and then CCR5, CXCR4 as the host factors. So this study that we are actually trying to understand is are there any other factors? Are there any other host factors that might be involved in the uh, regulation or in the aiding the HIV infection? Okay. So, well, let's see. GP41, this receptor that is present on the HIV, will interact with CD4 and also the receptor CCR5, CXCR4, and then it will make a stable interaction, and then there is a fusion between these two, and then eventually it will aid in the fusion of the envelope with that of the cell membrane, and then whatever the contents of the RNA that is in the HIV is injected into the cell, and then that's the establishment of infection. In the previous class, in one of the previous classes, we have also read about while this is the normal entry process and how infection happens, there are ways of, if we can block. From Berlin patient, we understood that if there is a variant that CCR5, if we knock it out or kind of mutate it, we can probably prevent the ability of virus HIV to infect the cell. So one could use inhibitors of CCR5. One could have antibodies against these CCR5, and also you can have chemokines that actually bind to CCR5. You can have smaller proteins. So that the idea is somehow to disallow the interaction of GP120, GP141 HIV's receptors, HIV's proteins with that of the CCR5. Okay. But then there are issues with that. Say if we try to have those kinds of something like this inactivating alleles, those people tend to have more susceptible. They are more susceptible to certain viral infections and cancers and many other diseases. So basically, if you are trying to go for gene therapy based approaches, then there might be some issues if you are making any inactivation of CCR5. There are also reported that these meddling with CCR5 can at the level of genes can have some problems within the individual. So the idea that is the basis for this paper. Are there any other host dependent factors that we can try to manipulate? Because CCR5, maybe that's all right, but it does have some issues. So if you want to really, and also there is a problem. HIV can mutate, and if it has mutated beyond its dependency on CCR5, then there is going to be an issue, because then that therapeutic approach will become futile. So one has to. So the idea is to find out more host dependent factors so that you can try other approaches as well, not only CCR5 based ones. Okay. As a part, using RNAi and many other types of screens, people have actually identified many different factors, host dependent factors, but there are conflicting or contradictory or the reports are actually contradictory, or there are some conflicts between what is right what is a host dependent factor for HIV and what is not. So the reliability is the issue there. And if in general, this is to recollect what we have already learned. Not only one is to stop the interaction of HIV with the receptors and co-receptors. To inhibit reverse transcriptase is one way. To reduce the ability of it to move into the nucleus is one another way. So at the different life cycle points, we can actually try to interfere so that we can either manage or eliminate HIV from the body. So the goal here, as I was describing in the past, in the previous slides, is that we want to identify the host, what are the host dependent factors. And here they are more into the protein level. Okay. And it's mostly about trying to see how trying to interfere with the infection. Okay, if the virus host dependent factors for infection is here, we are talking about HIV entering into the target cell. Okay, we are trying to find out host dependent factors required for HIV entry into the cell. Okay, so they have gone through this study happens to be built on many other sub studies that were previously reported. So the materials, if you want to really follow the cell line, what they have done and all those things, we have to read 10-20 other papers that they have cited in their methods. We are not going into all those details, but the essence of how it is done and what they have found is what we are going to focus on. Okay. So there will be some limitations here and there. You can adjust and try to read more if you want to. But my idea of this paper in the syllabus is how CRISPR can be used to find factors so that we can probably develop therapeutic strategies. There are also, CRISPR is also used in treating or eliminating HIV, or at least attempted to cure cancer, to cure HIV within the genome. Many other things have been done. So this is just another study in that way. So the first thing is they need to have a cell line. So before going into that, I would want to also say, what is a screen, right? Screen is something like this. You have so many cells, say you have 10,000 cells, and I want to pick one particular cell out of it. Okay. So you should have a screen here, something like this, so that you will eliminate all the 999 cells, right? And get that one particular cell. That is screening, right? So the screen here would mean that. So in this particular approach, what they would have done is they would have generated a cell line. And then in such a way, there will be some manipulation to it, which we will discuss in a while. And then they would have put in a screen where HIV is the screen. HIV infection, okay, is the screen. Those cells that get infected will be removed or eliminated. Right? Removed or eliminated. And the remnants which did not get HIV infection, not infected, HIV negative, those are the cells, something is, these cells are resistant to HIV. Right? They are resistant to HIV. Now, say, getting back to this one, in all these cells, they have made some genetic manipulation. And then they did some genetic manipulation. And then the screen is, as I said, the screen here is, I'm just using kind of symbolism here. It's not a screen per se. It is not a physical way. It is a strategic screen where the idea is you infect all the cells with HIV. Those cells that did not get infected, and then you study what is the manipulation in there. Then you know which gene is manipulated. Which gene is manipulated, then you know that that gene, because it is not there, and that would have been the responsible, that is responsible for giving this HIV resistance. Now we know then that gene protein, the gene product is actually the host dependent factor for HIV infection. If this protein is not present, HIV is unable to infect this particular cell. Okay. People in genetics use the word screen or genetic screen. This actually happens to be the logic of when you want to become a scientist. At least at one point, you should have designed at least one genetic screen in your career. Until then, we cannot be called as a scientist. In the sense, it is full of logic. You know what your output should be. If you know what your output should be, I will try to explain. Here, the output I'm expecting is cells resistant to HIV. I mean, once I know that cells resistant to HIV, what would mean then I will know about the genes responsible for it. This is my output. You are starting with a question. Then you should know how I can generate that. That will be your ability to generate this screen or design the screen. If I'm going to use a certain screen, something like here is to use HIV infection, then I should know what I should do to the cells, how to manipulate the cells. Right? You are not starting, majority of the research, you can always see it that way. They are in the reverse direction. You will do something, you will treat it with a compound, and you will see what happens. Instead, in genetic screen, the logic is something, you know what you want. Okay? And then you should know how to design a screen, and based on that, or on these two, how to manipulate the cells and how to design the screen will actually prove your understanding of the subject, and also your intellect in using and designing the strategy for it. So remember, try to find more papers on genetic screens and try to read them more, so that your ability to design screens will become much better in the future. Okay, let's move on. So the first thing, as I was telling, is this cell line. I will try to describe each step, cell line first, what they have modified, and then at the end and later time about the screen itself and the results. Okay, we are cutting it short actually. So they have taken a cell line which is CCRF. I mean, it expresses CCR5, and it also has hygromycin resistance. So I'm not going into all those details, but I will tell you what it has on its genome. So it has CCR5 gene, okay? And it also has, I'm not putting exactly how they are. I'm trying to explain the intent of it. They also have a HIV with GFP gene here, and a LTR, long terminal repeat of HIV. So you can probably consider it as promoter of HIV. Okay, just take it like that. So if there is a particular factor from HIV, it would have recognized this and would express GFP. And if you are looking at the, it will give green fluorescence upon, it will give green fluorescence. When will it, if HIV, that particular factor that can recognize this promoter, only then GFP will be expressed in higher concentration, and you will see bright green. If HIV is not present in the cell, then the promoter is not active in that situations, because of which GFP is not produced, because of which there is no green fluorescence. Maybe mild green fluorescence may be there. Okay, so I hope you understood that. It has CCR5 gene, and it has a reporter, that is the GFP, under the control of PHIV. For simplicity, I am saying promoter of HIV. Okay, so they have chosen, they have been doing multiple things, and then they chose one that is called, they named it as GXR Cas9. Yes, I forgot to mention that there is also a Cas9 gene here. Okay, Cas9, if you remember from CRISPR that we have learned in the previous classes, Cas9 is a nuclease. So it induces double-strand breaks if it is activated by the CRISPR RNA and tracer RNA, or single guide RNA, right? Here we only have Cas9. We don't have any CRISPR in the cell yet. So this is the GXR's Cas9 has three components that are relevant to this particular study. One is that it has CCR5. Okay, this cell line is based on CD4 cells. Okay, they have constructed a long time. They have been many people have been using it, I guess. So the three components are CCR5, and then they have a reporter for HIV, GFP, and they have a Cas9 gene also in the cell line. Now, before I go in, what I should want to say is, say for example, I have taken GXR Cas9. If HIV is infected, what color will it be? It will be bright green. Why is that? Because the reporter will get activated and there will be high expression of GFP. If a cell is there that is not infected, it will be it will not glow, or maybe it might have very mild green because of basal expression. Okay, so one should be able to separate those cells that are green fluorescing from those cells that are not fluorescing. In this line, we should know about a technique called FACS. I hope you have read it in somewhere, in some, especially maybe stem cells or somewhere. If not, we are going to discuss that a little bit in brief. So it's an important technique. It's not in the scope of this course, but I think it's all right it's important to learn this nowadays. So FACS is fluorescence activated cell sorting. Cell sorting, what does it mean? You have a bunch of cells, and then you're able to, based on some property, you're going to say large cells and small cells. You should be able to sort them, all the large cells in one tube, small cells in one tube. That is called cell sorting. And if you are sorting cells based on fluorescence, whether they are fluorescing or not, then it is called as fluorescence activated cell sorting. I mean, flow cytometry is another way. Here, this is a preparative technique. Flow cytometry is more of an analytical technique. Analytical technique, when you say, here it is preparation, right? Because you're sorting, and then you will use these cells for some other purpose, right? That is called preparative. If I'm just analyzing how many cells are there, I'm making a graph, how many are fluorescing and how many are not. So all those here, if I'm analyzing the contents of this particular sample, then it is an analytical technique. Okay. Most techniques are analytical techniques, right? Say gel filtration chromatography, you are separating proteins based on their size or so. Then if you're just directly analyzing what is the composition, how many proteins, which proteins are large, which proteins are small, you're analyzing, that is an analytical technique. If you are going to separate them into two, those that have high molecular weight from those that are small molecular weight, and you're going to use this for some other purpose, then it is a preparative technique. Okay, so the question here is FACS. In FACS, what they do is, I'll use a different color. Yeah. So you take a mixture of cells and you would have added antibodies against the cell surface markers with a fluorescence dye. You can choose, say, for example, there are 3 different cells. One cell has a receptor for this, another cell has a receptor for a green receptor, and I am putting another one for a red one. And I will choose one of the cells. Yeah, let's use it like this. Now I'm going to add antibodies against them. And the antibodies I'm adding 2 different antibodies. One that recognizes only this kind of cell, and I'm going to add a green fluorescence to dye to the antibody. So antibody is against the green receptor. And I'm also going to use another antibody, which is specific to the red receptor, and I'm going to have a red fluorescent dye conjugated to the antibody. Now, if I have this mixture of cells, I should make a single cell suspension. That means no 2 cells are aggregating. All of them are independent and they're buoyant kind of thing within the, or as a suspension within the liquid. Now, when that happens, they are allowed to pass through a fine capillary through this tube like this. That means only one cell at a time can pass through it. When the cell comes, a laser beam hits onto it, and if there is a fluorescent dye, it will emit fluorescence, and that is picked up by the detector and then sent to the computer. And the computer will decide. We will have used a program to it. If it is red, give it a negative charge. If it is green, give it a positive charge. We would have set to computer to do that. So if there is because the computer knows that it is red, and if there was, when it passes here, then if there is a green fluorescence, then it will give a positive charge added to it, to the droplet containing that single cell. If it is a red fluorescence, then it will give negative charge like this. And these droplets are allowed to fall through this tube like this. And here we have 2 electrodes. One is negatively charged, and the other one is positively charged. And all the negatively charged droplets will get attracted towards the side, and they will fall. Their path of fall is deflected into a different tube. And if they are positively charged, the droplets will be attracted towards or deflected towards the negative charged electrode that is here, and then they will be collected into a different tube. If we did not program that they don't have any fluorescence, what to do with it? We didn't do anything. Then no charge is allowed. Empty drops or any non-labeled cells, they do not get any charge, and they are not deflected by the electrodes, so they will fall into the middle tube like this. So I hope you see that we had a mixture of cells, and at the end we were able to sort them out. All red cells in one tube, green cells in one tube, and those unlabeled ones in one tube. That is called cell sorting, and here it is dependent on fluorescence. Our logic will depend on how we use this fluorescence. In this particular paper, these people already have green fluorescence within the cell, so you don't need to actually have add any antibodies like I described here. The cell itself can be in 3 different states, and those cells that do not have any fluorescence, those that they have little bit of fluorescence, and those that have high rate of fluorescence, right? So they should be able to, based on what you give into the computer, you can say that if there is this much intensity, threshold intensity of GFP, put them into one type of container. If there is no fluorescence, put them into another container, something like that. So you can separate them and find out, and then use it for your other purposes. I would also, because they also mentioned in this paper, and it's a closely related technique, I would also want to discuss about MAX. You may have heard about it. It's the same thing, cell sorting. Only difference is magnet activated. That means one, you would have used antibodies against using antibodies conjugated to microbeads, magnetic microbeads. So you would have you would want to say something like, I have cells, and somewhere else this is used, okay, not here. So if there is a receptor, you have an antibody, and there are two different types of cells. One has the receptor, the other one doesn't have. You take the antibody, you want to separate these particular cells with the receptors. So you add antibodies against it, they would bind here with the microbead, and that is what you have do labeling. Once you have labeled like this, you would allow, this is the column you see here, and you will pour all these cells here, and these blocks you see are magnets. So any cell bound to antibodies like this with the magnetic microbead, they will get retained in the column, and the rest of the cells will flow through. And once your flow through is completed, you also wash the column with to get rid of any cells that are not that are bound by, because of some other reasons, not the magnet. And then you remove the column from the magnetic chamber that you have, and then you elute the rest of the cells. So you had a group of cells of which only one was actually having a receptor, to which you have added the antibody with the microbead, and now you have all the cells would have been here in this case, and the cells with the magnetic bead will be separated or sorted out like this, and you can use it for your purposes. Yes, so let's get back to the screen. I will remind you before going further, so that they made this cell line. It is called as GXR's Cas9. They have CCR5 incorporated. It is there in the cell line. There is GFP under the regulation of HIV promoter. It is a different one. I'm using the term so that it's easy to understand. If there is HIV infection, GFP will be expressed highly. If there is no HIV infection, GFP will not be expressed. So GFP is the marker, the reporter here. And then they have also integrated Cas9 into it. It will be useful in the next strategies. So what they did then is they have used, now we are going to do with the CRISPR, right? What they have done is they made a plasmid library containing 187,000, these many number of single guide RNAs. Those say we have about 2,500 cells, or sorry, 25,000 cells, or say somewhere between 20 to 25,000 cells. Some of them are sorry, genes. If I said cells, yeah, genes in our genome, protein coding genes. Out of them, many of them are essential. Some of them are essential. If you delete them, the cell is dead. So these people have used, they figured out or reduced it to about 18,000, approximately 18,000 protein coding human genes. They made single guide RNAs for all of those genes, 18,500 genes. So, and then they also used some as some controls, which they used, there are non-targeting, some random, which do not have any specific sequences for any of the protein coding genes within the human genomes. So they made this, and they have cloned it into lentiviral lentivirus. And then the idea is you have the cell which is having the Cas9, right? And there are many other genes here. And so if I added a lentivirus with the sgRNA, I'll just put it in the shorter way. The sgRNA along with the Cas9 would form a CRISPR Cas9 complex, which will then go and target it. It will get inactivated. It will get cleaved, and then it could be it will then be repaired. And because during repair, what will happen is some of the nucleotide bases are eliminated, because of which this whole gene, although it will be repaired back, but the repair is not necessarily going to generate a functional gene, no functional protein. Right? Okay. So they have generated as many 18,500 types of sgRNAs. All the cells already have Cas9 in them, because we have used in the cell line we discussed in the little while earlier. And then they would have taken the GXR Cas9 cells. Okay, this is the cell line. You will take so many of the cells in millions, and the idea is to then add in the library of sgRNAs in the through lentivirus, and then see what happens. What are you generating? Then you would have different types of, all are GXR Cas9 cells. But there is a difference between cell 1, cell 2, cell 3, and cell 4. In each of them, a different gene um, is mutated. Okay? In each of them, different genes are mutated. If there is an essential gene that is mutated, that cell would die. Okay, they are not bothered about that part. They will only grow all those cells that are able to grow. Those cells that are because of the mutation, if it is lethal, they would die. Let them die. So now they generated a large cell pool. All of them, the base is GXR Cas9, but there is one single gene mutation in each of them, unique single gene mutation in all of the cells. Okay, that's the difference between the pool within the cells of this one single pool of cells. If you are losing me while I describe it, don't worry about it. Again, I'm going to repeat in one slide summarizing everything. So probably you will be able to find out. Without understanding the intricacies of what they're doing to the cells and the CRISPR, we wouldn't be able to understand the genetic screen. Okay, now that is how they generated a pooled genome-wide CRISPR screen. That means they have a large number of cells. Each of them have a mutation in any one of the 18,500, approximately 18,500 genes or protein coding genes. Right? Now what they would do is, what they want to do is to generate or add in, you already, so wait. We have the cells, GXR Cas9 cells. Then we have put in the pool of sgRNAs, 18,500. Then we now have several GXR Cas9 cells, each of them in one cell, gene A might be inactivated, in another cell, gene B might be inactivated, in another cell, gene C, and so on. Now we are ready with the material for screening. And here we will do the screen. What do they do with the screen? They are going to add HIV. They're going to treat all these cells with HIV. So what may happen is, because of HIV infection, some cells may die. HIV-induced death. Right? And what else may happen? if there is something else, right, in GXR Cas9, what are the three components that we described? They have CCR5 gene. They have Cas9 for using in this step. They also have a reporter gene, that is GFP. And that reporter gene will express if there is infection, this promoter will be activated and GFP will be expressed. So some of the cells are GFP induced death, so they would die out. Those, and then what we will have is all those cells. Some cells will have high GFP expression. Some cells will remain same, low or no GFP expression. What does that mean? What does it mean to have high GFP expression? What does it mean to have low or no GFP expression? If it is high, that means HIV has infected. And low or no GFP means no HIV infection. Why did some cell have high GFP and some cells have low or no GFP? What is the variation in all these cells? In each of them, you may have different gene inactivated. Say, for example, I will just call them as cell A, B, C based on their gene of infection. If I said cell A, cell C, and cell D have high GFP expression, and cell B, cell E have low GFP expression. Okay, these have high GFP expression, and B and E have low GFP expression. What does that mean? It means, why is GFP high? It is because HIV infection has caused the expression or overexpression of GFP, and that's why it is high. That means the mutation in this cell, right? You mutated gene A, and yet HIV is able to infect. That is what this means. What about C? The same thing. Even though there is a mutation in gene C, and still HIV is able to infect. That means that gene C is not involved in the uptake or in the infection of HIV. And that is why it is having high GFP. It is surviving and it is having high expression. High expression indicates it is HIV infection. The same is the case with gene D. That means it is allowing, even though there is mutation in gene D, still there is HIV infection. Right? That's what it would mean. If we look at B and E cells, what does it say? It says gene B is mutated because of the single guide RNA that we added, and because of which, and it did not get HIV infection. HIV has failed to infect it. What does that mean? It means that gene B is directly or indirectly involved in the uptake of or in the infection of HIV. Okay, that is what it would mean, gene B and gene E. I hope you understood the screen of this. So based on that, what they found is they've got some cell lines, and in all of them they did sequencing, of course, which I'm not going to discuss in general, but they know the sequence of single guide RNA, because of which you can always figure out which gene, where is that particular mutation that is present. And then they figured that there are 5 hits they got: CD4, CCR5. We already know these two, right, from the beginning of in the background that we discussed. CD4 and CCR5 are essential through which the HIV causes infection. There are 3 additional genes that this screen has found out: TPST2, SLC35B2, and ALCAN. Okay, what they are and how do they function is not necessarily for our understanding of rDNA. If you want to look into HIV, try to find out what is it doing in the infection, then it is fine. That's the knowledge part of it. It's not necessarily our technical part. So I will summarize once what these are, and then I will give a brief introduction about these two, so that you can probably read upon it later. So I'm reviewing what I have discussed so far. Please try to follow, and if you have problems, you can always ask questions. So this is the actual cell line they have taken. That is wild type CCRF SEM cells. Then they have put in the PHIV GFP, that is for reporting of HIV infection. That's why they have used something like this, a promoter with GFP, and this promoter is PHIV. You can talk of it as like that, simple. And then CCR5 is there. They have incorporated the CCR5, and then they have Cas9 also integrated. Those are the three elements that we require. CCR5 is kind of essential, or it is essential for the type of HIV strain they have used. So it is CCR5. Cas9 is for making the mutations in the later step, like here. So then they have selected, after doing all this preparation, they have now came to GXR Cas9 cells. GXR Cas9 cells have a reporter gene with the GFP like this. CCR5 is put in there, and also Cas9. Then they have done the mutagenesis. Mutagenesis of all the genes, all protein coding genes. Okay. In that, they have chosen about 18,500 protein coding genes. They have also, and they have taken in excess, in the sense, at least 1,000 cells per single type of single guide RNA specificity, to increase the probability of representation of mutations in all the different types of genes. So they have taken, because they want to increase the probability, they have taken a large pool. They made 1,87,000 of ssRNAs targeting these genes. And then they have done the mutagenesis, and then they have pooled all the mutant knockout collection. So in all the different cells, one or the different other gene is inactivated or deleted. Not deleted, or inactivated is the correct one, because it makes a single cut, and after the cut, the non-homologous end joining will happen, because of which the gene is usually mutated. So now we have a question is, this is the pool that is select cells. Now they have used HIV, which is CCR5 tropic. That means CCR5 is essential for it, which gains entry through CCR5. They have infected multiple times for about 6 weeks. That is the HIV challenge. So all those HIV induced deaths, cells that have died because of HIV, would be eliminated. Those that have high GFP cells, high GFP expression, what does that mean? That means HIV as infected. So those cells are also sorted and eliminated from the pool. Now what we have is a surviving mutants. All of them would have GFP expression, no GFP expression, or low GFP expression. What does that mean? It means that HIV has not infected, no infection of HIV. So somehow these cells survived HIV challenge, and that survival is because of the mutation present in that particular cell. And now if we know what gene is mutated, we can figure out that that is the host dependent factor for HIV to gain entry into the cell. Okay. They have of course used sequencing to compare the sgRNA and many other things like that. So as I said, obviously they found CDR4 and CCR5. As if these genes are mutated, what would happen? Then HIV cannot interact with the cell to, and thereby gain of entry is prohibited. 3 additional genes are also found, and I will give briefly discuss them so that, and then we will end the class. So there is these 2 genes are involved, TPS T2 enzyme and also SLC35, uh, SLC35B2. CCR5 has sulfation. I mean, several residues of it are actually sulfated in the Golgi complex. This is the Golgi complex, okay, here, and this is the cytosol. So within these 2 enzymes, TPS-T2, that is tyrosyl protein sulfotransferase 2, okay, is required for sulfation of CCR5. There is also one more, that is it's a transporter in the Golgi complex, the SLC35, that is the other one, which is important for transporting phosphoadenosine phosphosulfate into the Golgi complex. If this is not there, then even if this enzyme is present, the substrate is not present. So sulfation of CCR5 would not have happened. And apparently now what we know is once this one is matured, it will be secreted out, and there will be a lot of sulfated residues like this. If there is no sulfation, then HIV is unable to interact with it. That is what is known from this study. So if we have inactivation of TPS-T2, no sulfation, and that is conferring resistance to HIV infection. Similarly, also SLC35, if we do not have this, there is no import of PAPs, and that is the substrate for sulfation of CCR5. And because there is no substrate, there is no sulfation of CCR5, and then also it is resistant. The cell becomes resistant to HIV infection. Okay, and the last one is Alcam. So this is something new for me too. So HIV can transfer between cells. It's not always that HIV has to infect and get in like this. HIV somehow can also move between the cells. There are viral synapses that are formed, something like this. There are multiple ways where the cells form clusters. They interact. By the way, Alcam is a protein, cell surface receptor, cell surface protein, which will allow the binding of different clustering of cells, and that is going to increase the probability of HIV virus from one cell going into the HIV into another cell. So what these people have found is if this particular gene Alcam is not present, then the cell interaction, the clustering of the cells is minimized, because of which the HIV spread from cell to cell is prohibited. Okay. So there are several mechanisms you can study if you want to, but I think as I was trying to emphasize in the beginning of this class, please know how genetic screens work. Please try to, first thing, as a student, our job is to understand how people design these genetic screens. What is the objective? What did they prepare? What is the logic of the screen? Right? It is a thinking process. It's highly intellectual. It's not necessarily about it's not like just sequence everything and see what happens. It's like you only want to do, it's a smart science basically. So you should spend more time trying to read about genetic screens. If you just type genetic screens in PubMed, you will get many papers. Start with the smallest, simplest ones. I should admit that there are some screens that I don't understand too. That means I will have to spend a lot of time to understand them. If you're at your age, if you're trying to do those, I think you'll be a fantastic scientist in the future. Okay. I hope you understood. If not, you can always come back and we can always discuss this again. Yes? Any questions? If not, we will discuss in the later classes. Time is up, I guess. Okay, take care and have lots of fun. Read the paper and please get back. Thank you. Bye-bye.